So the heart itself is a very reliable pump. It beats about once a second for every second of an entire human lifetime. Now here in this cross-section, we see that the different cells, or the specialized cardiac myocytes, have different electrical waveforms. Going from the top to the bottom, we see that as we go to the more muscular ventricles, corresponding to the strong outflow required to distribute blood to the entire body, we have this prolonged action potential. There's a depolarization corresponding to the initial contraction, and then a recovery. And then this, in aggregate, corresponds to the ECG, or what you might see in a hospital, or here in a hospital or a lab, the rhythmic beating corresponding to the activity of the heart. Now, taking a closer look at this action potential, we see that it has multiple phases. Now, this emergent phenomenon has underlying ionic components. Namely, this upstroke, or when the heart initially contracts, is due to the sodium moving into the cells, and is the balance of calcium and potassium that leads to this gradual recovery. Now, in terms of the biophysics, the actual structure of these channels, these voltage-gated ion channels, lead to the actual dynamics you see here, namely when the channel opens, what ion it's selective for. But in terms of a physical model, what we have is fundamentally the movement of charge. And here the model formulations come as a circuit diagram. The cells, they separate charge. We have a capacitive term. We have parallel pathways for the different underlying ionic components for sodium, for potassium, for calcium. And in terms of how these models are structured, they're constructed branch by branch, as in experiments are done on a single ion channel, and it is the goal of these models to integrate that information and provide useful insights into the biophysics, and also whether someone has a chance of proarrhythmia, or it might be cardioprotective. And in terms of what these parameters mean, for example, this maximal conductance here corresponds to the number of channel, namely how high this peak is, or what the expression level is. So our goal in this study is to relate the underlying ionic components to what is going on in the overall electrical behavior. And so in working toward this goal, it's motivated by the limitations of generic standard models. So for example, there's natural variability between all of us. There's different configurations of our proteins. So for example, this trace here, in even a simple comparison of the action potential duration, which corresponds to the important pumping function, we have variability in terms of its duration, namely when the heartbeat ends, comparing two cells here and the generic model. In terms of how these models are built, they're done under different protocols, and they're also done via separate underlying components. There's another challenge of the anatomical ambiguity. When the models are built, it's not necessarily clear which specific location in the heart it's coming from, namely where in the heart wall. And in particular, and what's been the focus of this body of work, is we want to move away from these hand tuning of parameters to get the certain electrical behavior. So what we see here is an example of different parameters being pushed up and down, leading to a certain response. But that can only go so far. Two different models can match an action potential duration, but in terms of another important behavior here, calcium, there's a small mismatch. So in terms of how do we search this parameter space, so even if we consider a subset of the total parameters in these cardiac models, we have a combinatorial explosion. The brute force approach won't work. There's also another important challenge in terms of the compensation. Here we have Rastigan's test function, whereby two parameters or subsets of parameters can compensate for each other. Now this is useful in physiology. For example, if one person has a mutation, one of their potassium channels is not able to function properly, the cell will compensate and overexpress a related potassium channel, so the relatively normal function of the heart can continue. So the approach we used to traverse this large and multimodal parameter space is a genetic algorithm, and Carolyn Phillips touched on this, this method a bit yesterday. And so in terms of adapting this approach for this problem, we went through a number of subroutines in regard to selection, crossover, mutation. I'll just show an example here. So what does it mean to combine two models of a certain form? Here, with the same model structure, but different parameter instantiations, as in parameter combinations corresponding to those currents, their peaks in particular, we have parents that are selected based on their overall fitness, namely error away from your target behavior you're trying to describe. There's a crossover operation, namely parameters are inherited from each parent, and also mutation to add further diversity and progression of the genetic algorithm, until we start filling the next generation. So what does this look like in terms of our actual problem space? So red is our target behavior. These blue bars are up and down of the corresponding parameters. And in our first generation, we have correspondingly high error. Namely, we have a mismatch. And then as we progress from generation 2 to the completion of the optimization, we have both a reduction in the generational average error and the best individual error until we have a solution that matches our behavior. So with this approach, we were interested, what protocols can we use that are efficient in time and in terms of accuracy to get an estimate of the underlying ionic components? The electrical behavior is an emergent phenomenon, and we're trying to understand what's driving it. So the models themselves are designed to match this sort of static rhythmic beating. Here you see a train of action potentials. But a very rich set of dynamics happens during arrhythmia, namely where the heart begins to beat faster and particularly quiver. And unless there's an intervention, this can be very life-threatening. So here, we developed the approach involving stochastic pacing to sample the dynamics, where instead of having the traditional static train, we're applying with random diastolic intervals. So what that looks like is here, where we're having long beats followed by short beats. And in terms of the part of the dynamics this explores is that as the beats get faster or you have closer beats one after another, you have a shortening in the action potential duration. So for example, this is shorter than the previous action potential. And this is important, for example, when you're running or exercising, your heart needs to beat faster, there needs to be compensations. So in terms of using this set of dynamics as an objective function in our genetic algorithm, we're able to show that there's a fit in terms of the electrical behavior. What we were surprised to see is that comparing the overall parameter estimations, a perfect estimation would be zero, error bars around zero very tightly, that the stochastic train compared with the single action potential train compared with another protocol, and in terms of the lengths and complexity, we have the comparisons here, the parameter estimations are comparable. I have the quantification on the different slides. But what we were also fascinated to see is that with the stochastic pacing emerged a predictive capability. Namely that when we tested on novel sequences, we had the lowest error of the three. Namely comparing against novel stochastic pacing sequences, static pacing sequences, and another form of long-short testing sequence. So the thought here is that there's a generalization, namely that the parameter space has been sampled and we're able to predict a larger body of the possible sequences. But we still were not satisfied with the overall underlying estimate. After all, we want to know in a specific individual, from one individual to another, which proteins are up and down. This affects which drugs might be prescribed. So we sought then to reverse it, to instead of looking at the voltage, to look at the current response in the circuit. So here we have different voltage levels, which the cell is held to, and the underlying current response. And here, the protocol was designed to isolate the different components corresponding to the biophysics. So for example, one of the potassium channels is activated below negative 100 millivolts, while sodium is closer to 0 millivolts. So what came out of this analysis and development, that a prediction that those ion channels that were well represented, namely that they had a dominant component in some part of that voltage step protocol, the genetic algorithm should be able to use that information, and we should get very good estimates of the particular parameters, in this case IK1, INA, IKS. And indeed, this is what we saw when we revisited the parameter estimation problem. The parameter associated for sodium here, we've been able to estimate the underlying parameter also for IK1 and IKS, with varying degrees of success for the other parameters. Also, excitingly, we're able to show a drug block case, namely estimate a very different state of the model when an actual subset of the channels is blocked. So in looking at this analysis, going from the emergent phenomenon of the electrical waveform, the underlying ionic components, and using a computational approach to bridge the two, we were able to estimate the underlying nominal model, and this was mediated by boosting and shrinking in an automated way the underlying ion channel conductances. We've also been able to determine some predictive power, also useful to test different drug configurations, and also looking at the addition of the voltage step protocol, how we can produce cell-specific and individual-specific models. So I would like to thank the lab and very sincerely thank the DOE CSGF and its supporters. I'll take any questions you may have.